Mental disorders have a genetic underpinning. That means that there are some genetic factors, risk factors, variation of genes that actually increase the risk. Most of them don't make you ill, but they increase the risk that you may develop a mental illness. What we didn't know is what happens in the brains. We knew that the brain is much more complex than we anticipated. There are many more cell types. There are different areas of the brain that likely have different functions, and some areas are more likely to be more involved in the expression of a mental disorder than others. So we decided to start this program that we called PsychENCODE. Let's look at the brain specifically, and let's see if we can link genetic signals to genetic regulation, to molecules in particular regions of a brain and particular cell types, and let's see if we can associate that with disease. And guess what? The PsychENCODE project came through, and indeed we are now seeing the first signs of molecular perturbations, we call them, changes in particular cells, in particular cell types, in particular neurons that are associated with mental disorders. We are at the beginning. I'm not trying, I cannot overstate how relatively early we are, but I can confidently say for the first time we have a beginning of an understanding of the biology, the molecular pathophysiology of mental disorders, schizophrenia, bipolar, and autism spectrum disorder. What we also learned from this first pass of this initiative is how important developmental timelines are for brain development and the risk for mental disorders. So there is a temporal trajectory that can be changed by genetic perturbations, and those in turn increase risk for mental disorders. Some of the projects in PsychENCODE actually deal with cellular model systems. Because it is so difficult to study the brain directly, what the scientists have been able to do is to take tissue samples, they may just take skin tissue and turn skin tissue into neurons, into brain cells, and then characterize those brain cells, those derived brain cells, phenotypically and molecularly, and to see whether disease signatures are preserved. A bit surprisingly, but very interestingly, in PsychENCODE we find some traces of these of disease signatures in cellular model systems, so sort of opening the door wider for the use of cellular model systems as models for neuropsychiatric disorder. We actually see perturbations in gene expression networks that can be linked to specific diseases, for example, autism. So with the PsychENCODE project, we were actually able to identify several hundred new risk genes for mental disorders, in schizophrenia in particular. There's a study in PsychENCODE that makes comparisons between primates and humans, and they were actually able to identify human-specific genes that are expressed across development that are risk genes for schizophrenia. So there are specific genes that are unique to humans, and these unique genes are involved in the pathogenesis of mental disorder.
This needed concerted effort. Many investigators had to come together. I had to do this collectively, and that's where they get enough number of brains and samples to make scientific inferences, and that's why we formed this collaborative consortium for them to share in real time the data that they generate. They have provided us some biological insights into what could be the critical time windows during development of the brain where these genes could influence the disease process. There's no single gene that contributes, and its polygenic nature is also seen at the transcriptome level, and the regulation of this transcriptome is globally impaired. So the RNA or the gene expression changes on a global scale. They built these computational models to predict resist much better than the previous ones. So by integrating this data across different levels like transcriptome, epigenome, proteome, and regulome, they are able to build much better predictive models of the disease. So when you combine all these sets of information in a computational method that learns from these different data sets, this learning model tells us about the likelihood of that disease occurring when you have this particular variation at each level. By comparing the readouts, gene expression readouts, which is transcriptome between schizophrenia, autism, bipolar, from cross-disorder analysis, they could see convergent molecular pathways. So these are distinct diseases, but there are some aspects of diseases where the biology is similar. There are network modules. The genes interact with each other in a way to influence the disease process. If we can find clues early on, biological clues, we can intervene early on. While we are building and generating more data, analyzing this data to find basic mechanisms, there's an opportunity also for drug discovery.